Okay, we'll get started. Thank you everyone for joining this month's Funk Lecture. We're super excited to have Mario Moura present his work today. But before the talk begins, we have some announcements um, from the IBS. Our biennial conference will be in our house in January of 2026, and currently abstract submission is open and will continue until September. And then beginning in November, standard registration will begin. And yeah, at the end of the talk, we'll have a Q&A. If you have any questions, you can either type them in the chat or raise your hands and we'll unmute you and you can directly ask your questions. Yeah, and again, thank you for attending. All of our talks are recorded and will be posted on our YouTube channel at some point in the future. And for our next seminar speaker, we will have Brian McGill. And I will pass this to Marta to introduce our speaker.
Thank you very much, Raymond. Just one thing I would like to add is that the next seminar, Funk seminar, will be in September. We'll take a little bit of a break for August. A lot of people are on vacation, so we'll let folks enjoy the summer if they're in the Northern Hemisphere, and winter, I guess, if they're in Southern Hemisphere. But yes, the Brian McGill's talk will be happening in September. So please make sure to subscribe, register for that, and come to what will be quite possibly an amazing talk. But today it is my very pleasure to welcome Dr. Mario Moura to our monthly Funk lecture series. Like Raymond mentioned already, my name is Marta Jarzyna, and I'm the Vice President for Public Affairs and Communications at the International Biogeography Society. Dr. Moura is a professor at the Federal University of Paraíba in Brazil, where he leads the lab of biodiversity, ecology, and conservation. He obtained his PhD from the Federal University of Minas Gerais in Brazil as well. And then he held several postdoctoral positions, including one at Yale University, which is where I met Mario for the first time. And we've been colleagues and friends ever since. Dr. Moura's lab investigates the drivers of biodiversity patterns and extinction risk, combining biodiversity data science, spatial analysis, and phylogenetic methods. His work spans conservation biogeography, macroecology, and global change biology to assess biodiversity responses to anthropogenic pressures, with the overarching goal to reduce knowledge gaps and improve predictions of species vulnerability for conservation planning. So it is my absolute pleasure to welcome Dr. Moura to our series, and without further ado, Mario, the floor is yours. Feel free to share your slides as soon as Raymond stops sharing his.
Hello, everyone. Are you listening well? Yeah?
Yes, but we can see your
my presentation
presenter.
Okay.
Yes, this is perfect. Great.
Okay. So first, I would like to thank the organization for this invitation. So I'm really happy to be here and be able to talk a little more about my research. Sorry about my rusty English, perhaps. I'm trying to do my best here. And today, I'm going to talk about uncovering the blind spots in biodiversity data. It's a huge team of research in my lab, and I'm actually going to talk about biodiversity knowledge shortfalls. And when we talk about these knowledge shortfalls, the first thing that came to mind is how many species are there? That's one of the most direct questions that we have. And so far, we know more than 2 million of species have been described on the planet, but this number, it actually represents less than 10% of all living species. And how do we know that? How can we quantify the unknown? How do we go from 2 million of species described to 20%? Actually, most of the previous works trying to deal with this question, they were using species discovery rates, extrapolating the behavior of those species discovery rates until they reach a range of 0 species described per unit of time. So you can access how many species you are describing, at which speed, at which rate, and understand how long it will take to describe all species and how many will be described in that period of time. It's by extrapolation of species discovery rates, or sometimes by using species accumulation curves. This map that you are seeing at the screen, it is showing how species richness patterns are changing through time as we accumulate more knowledge on the geographic distribution and description of species. But something that it's really obvious in this map, it's how discovery rates are varying across space and time. But this map, it's not showing another important dimension that we need to consider when talking about species discovery, and that's the taxonomic one. So we have here 2 species, Rhea americana on the left side, the greater Rhea, locally known here in Brazil as Emma, and Brachycephalus darkside on the right side. So Rhea americana, it is the largest bird in South America, and Brachycephalus darkside, it is a really tiny frog. The adult has less than 1 centimeter. It's among the smallest vertebrates in the planet. Rhea americana occurs across most of South America, and Brachycephalus darkside occurs in a single mountain in southeastern Brazil. It's really endemic. So one species was described in 1758, the other was described in 2017. So there is a clear influence of biological attributes here, affecting the chances of describing this species. When we use species discovery rates, the previous works, we are considering all species with equal chances of discovery. So they do not have those equal chances. And it's necessary to incorporate how these species level attributes affect our estimates of unknown species. And this was the goal of this research. We published it a few years ago. And here we put together a huge database of species level attributes and try to derive metrics of species discovery potential across major clades, regions, biomes, and countries. And how we did that? Actually, we applied time to event models. This kind of analysis are also known as survival analysis. They are pretty common in medical and social sciences, like time to die, time to recover from a treatment, time to get a job. Here, we are assessing the time to describe a species, the amount of years between 1758, the beginning of the modern taxonomy with Linneo, and the year at which the species was described. And we have several different attributes. We have body size, geography. Here we have indicators of range size and also range rarity, if a species is co-occurring with other endemic and rare species. Accessibility is measuring human population density at the year in which the species was described within its range. And also distance to remote, distance to major cities. Taxonomic activity is measuring how many taxonomists were working with a specific clade in a specific moment in time, or how many taxonomists were working in a specific region in a specific moment in time. And also environmental characteristics, like if a species occurs in a warm or cold region, in a dry, wetter region. So after running these models, what we get, it's an output that is a set of values. Each one of those curves, it's the output for one species. So we have a set of values indicating how the probability of description is increasing across time. If I show this with traits, we have on the left side an example. Here we have 7,000 discovered curves for amphibians. And I just colored those curves with the color of range size. So we can see that orange curves, they rise up really earlier, indicating that the description of those species have higher chances, like species with wide ranges. In contrast, we have narrow range species with the curves rising really late, indicating a more difficult here to describe species that are narrow ranged. So after computing these curves, our goal here is to understand how to get an estimate of the number of unknown species. To do that, we need to get the discovery probability of each species in the present time. So we go here to 2015, that's the present time in our study, and we extracted the information of discovery probability, a single value for each species. And that behaves like a new trait that we can explore using traditional macroecological approach. By averaging that discovery probability for a species in a sample here can be a clade, a region, a grid cell, we can get the proportion of known species. This is the average discovery probability of all species in a sample. We know the species richness of that sample. We just need to count the number of species. And then we can just apply a rule of 3 here to get what is the estimated total richness for that sample. By total richness, I mean the number of known species plus unknown species. If we do the subtraction here, we can have the number of unknown species estimated for a sample. And we did that across grid cells and also clades and other stuff. But here I'm showing to you a map of the overall pattern across all tetrapods. And you're seeing some grid cells contoured by black lines. Those are the top 10% grid cells with highest number of unknown species. These top 10% grid cells will harbor about 70% of all future discoveries in the planet. And tropical broadleaf forests, they will harbor about half of all future discoveries. So this is just to show these species discovery hotspots, and of the discoveries that will be happening in the future, about half will be of reptiles, 1/3 of amphibians, followed by mammals and some new birds still being described. We can redraw this map using country borders. So here we have pie charts proportional to the number of undescribed species estimated for each country. And what is cool to see here is the top 4 countries emerging on the map. So Brazil, it is came in first with 10% of all future discoveries. Colombia, Indonesia, and Madagascar come next. And these top 4 countries together, they will harbor about 1/4 of all future discoveries. So this research was really cool. I was really excited when doing it. And it was targeted by a comment published last year. So in the comment, Buttel and Borzay, they questioned the use of species accumulation curves and model assumptions related to that method. But we did not apply that method in our research. We actually applied time to event models. And we took this opportunity to clarify our strategy, the assumptions of our models, and also to perform an independent validation. That was where I would like to go. So in that previous research, we trained our models using species described between 1758 and 2014. We did not include very recently described species because in order to train our models, we need to have spatial information available for each species. If a species was described by less than 5 years, we considered that the geographic information available for that species to be poor, not be enough knowledge on the geographic distribution of that species, and we did not include it in the model. So that's why we only worked with species within this time range. So for the reply, we actually traced it, we searched for all discoveries that happened in the subsequent years, and we tried to compare what were the proportion of discoveries expected within our top samples, the top clades, top biomes, top countries, top regions of grid cells, what was the proportion predicted, and what was the proportion that we actually observed based on the discoveries that we identified during this period. In the reply, we aimed for amphibians, so we end up assessing 1,200 discoveries of amphibians. Each cross that we are seeing in the map, it's a type locality of a newly described amphibian species. And here on the left part of the map, you are seeing a fraction that is the result of the observed proportion of discoveries observed within top samples. By top samples, I mean top 5 suborders, top 30 families, top 30 countries, top 5 biomes, and top 10% grid cells. So that's the observed proportion within those samples divided by the estimated proportion. If the value is above 100, it means our projections were performing better. If it's below 100, our projections are just close to that. And that's what we saw here for the last two patterns. That's because we are assessing, we are performing this validation using a single point locality data for species, for each species. We do not have knowledge here on the geographic distribution of this newly described species, just the type locality information. If we were able to improve the knowledge on geographic distribution, probably these two values would go higher. So if we expand this to all tetrapods, this is some unpublished results. Research in preparation. So using now 3,000 species descriptions of tetrapods, we can see that our estimated portion of future discoveries per biome, per country, per family, across higher level taxonomic ranks, our estimated portion matches really well the observed portion in the y-axis. So this was a really comforting result to see how well those estimates were approaching what we have been observing during the last decade. When we map those discoveries, we can see that we have all type localities here mapped for all these species. And we can see that our projections, they are denoted by this black line here in these radar plots on the top. And we have the observed values of discoveries within the top samples in colored lines. So if the colored line is overlapping with the black line, it means we are doing really well. And that's happening for most of the top samples, except for the top 10% of grid cells. But again, we are close to that performance. We just do not have enough information on the geographic distribution of those species to properly access the influence of these top 10% cells. But overall, this research helped us to show where we can put more boots on the ground to discover new species in an efficient way. But what happened to those specimens after we collect them in the field? Now we have a different opportunity. Most of you might already suspect those specimens will be housed in scientific collections. When a specimen is collected in the wilderness and reaches a scientific collection, it needs to be properly examined by a taxonomist. If that specimen belongs to a new species, it's a new taxa, it might become a type specimen that will be the critical reference material for a formal description. Like the most important type specimen, it is the holotype. And these last 4 steps that I just talked here, they might take a while. It's not something that happens really fast. So there was a study in 2012 that assessed the average shelf time. Shelf time, it's the amount of years between the holotype collection date and the date of description of the new species. And the average shelf time was 21 years across different sets of organisms. So you can see that this study aimed in very different groups. But for tetrapods, the average shelf time was estimated as 30 years. The study was based on almost 600 species discoveries, but for tetrapods, we have 11 species only. So we took an opportunity here to actually inspect what are the drivers of shelf time among reptiles. So reptiles are the largest group of vertebrates where most of the discoveries will be happening. And nowadays we have 4 million of preserved specimens of reptiles housed in scientific collections, 12% unidentified, and therefore a huge potential here to some of those shelved specimens be representatives of unknown species. So in this research, we reviewed about 2,400 descriptions of reptiles tracking information on 9 biological and sociological factors. We also use the time to event models here. It's a same framework. We are doing both research in parallel. And the difference here is in our x-axis. Instead of assessing the number of years between 1758 and the date of description, it's the number of years between the collection of holotype and its description as a new species. So here, a map just showing where are the type localities of these 2,400 reptile species. You can see type localities for lizards as orange symbols and for snakes as the blue triangles. And the tiny box plots, they are just showing the median shelf time for lizards and snakes in each biogeographic realm. We can see that overall, it seems that it takes more time to describe a snake than a lizard. We found a median shelf time of 5 years, way below 30, estimated previously, with a range of 1 to 155 years. And at least 25% of all reptile species were shelved for 12 years. So this is the upper quartile, 12 years of waiting for those species to be described. If we expand this for all tetrapods, this is also unpublished. We are talking now about 8,000 species description. We have a median shelf time of 5 years, ranging from 1 to 182, and an upper quartile of 11 years. And what were the drivers behind these patterns? So here are the predictors that we use to access the time to event models. And you are seeing these different colors and symbols because some of our predictors, they actually have some time constraints associated with them. For example, collector is an author. This is a binary predictor. It's informing the presence or absence of the holotype collector as an author of the species description. So if a specimen was collected 150 years ago, it will be pretty difficult for the holotype collector to be an author of that description. Right? In many cases, either the collector already retired or they might be dead. So that's a thing that we need to consider in these models. And this also happens with molecular analysis. A specimen collected 100 years ago will be really difficult to that specimen to have DNA extracted, since the technique was not available back then. Of course, we have novel techniques nowadays, but we did this sensitivity analysis restricting the size of our data set to include the species collected in the last 50 years, 55 years, and so on. We can see that the effect size of the predictors, they remain consistent. We have some quantitative differences, but not qualitative differences. And what are the major conclusions that we can get from this? So we need to verify specimens collected by non-taxonomists. When a taxonomist go to the field, he or she, they might collect a specimen already suspecting that it belongs to a new taxa. So they will go to the lab, properly examine that specimen, and move forward with the publication of a formal description. But when that specimen is collected by a non-taxonomist, that specimen may remain hidden in scientific collections for several years. That's because most scientific collections do not have an expert on every kind of organism that collection is housing. So those specimens, they will be properly examined only when a taxonomist visits that collection. That's why taxonomic revisions are pretty important. So
It looks like we have some technical difficulties and we might have lost, well, we certainly lost Mario. Let's give him maybe a couple of minutes to join back in. There he is. There you are.
Okay. Yeah, I lost my connection.
Okay, well, good.
Let me start again.
Yeah, go ahead. For a second, I was wondering whether it's on my end or your end, but I'm kind of glad it's figured out now. So go ahead.
I'm back.
Oh no. Are you still here, Mario?
Yes. Hello?
Yes, I can hear you.
You can hear me?
Yes.
My internet connection is unstable. Oh, come on.
Well, I can still hear you. Can you hear me?
Yes, I can hear you.
Okay. Well,
But are you seeing my slides?
I can see your slides. I can hear you. I can see you. So let's just
keep
going.
Let's move on. So finally, I was talking about the need to look at less specious genera because species-rich genera, they have lots of descriptions available. So that's like having multiple pieces of a puzzle. So you know how to diagnose, how to distinguish species from one another because there are several works available out there indicating which traits you need to look in order to distinguish species. And that's not something that it's happening for species poor genera. So species poor genera are more likely to have unknown species hidden in scientific collections. So far, I have been talking about how can we boost discovery of new species, either in the field or inside scientific collections, but we can also talk about other types of knowledge shortfalls. When we think about species that we already know, those that have been described, there is one type of information that is pretty basic and sometimes it is not available, and that's natural history data. So in this paper published last year, we try to quantify our knowledge shortfalls in natural history data, but we also try to fill those gaps. We took advantage of some fully sampled phylogenies available for major groups of tetrapods. We compiled information on natural history data and then applied a machine learning method, XGBoost gradient, to input what will be the expected value for each missing data entry in our database. So in doing so, we were trying to understand what is the invisible fraction of the trait space of the natural history data. Overall, our database was composed by 70% of data papers already published. We incremented those data papers with about 10 to 15% of new records compiled by us. And the inputted data, they were about 20% of the information of our database. So these inputted data, they are representing the invisible fraction of the trait space. And what happened with biodiversity patterns when we make those invisible fractions visible? So that was the next question that I would try to access in that research. We were actually mapping biodiversity patterns across space and across clades using those natural history traits. Here we have average verticality. What is verticality? It's a score that we derived from microhabitat information. A zero value here means a species is strictly fossorial, and a value of 1 means a species is strictly arboreal or aerial. So we have the observed pattern, the average verticality value per pixel using just the observed data. And we repeated that information using observed plus inputted data. This is a fraction, right? This is math with maps. When we get the result of this fraction, we can understand where on Earth we are missing more information on fossorial species or on arboreal species, or where that information is missing at random. So when we computed these maps across all traits, this is what we got. So the top two maps, they are showing body length and body mass. And the bluish pixels, they are actually indicating regions where we have more missing data for small-sized species, species with small body length, species with small body mass. And the bottom map about activity time, this is also a nocturnality score, where 0 values means a species is strictly diurnal, and 1 means a species is strictly nocturnal. So we are seeing these reddish pixels indicating that we are missing more information about nocturnal species. So this was a pretty cool way to identify these invisible fractions of trade space. In this research, we also highlighted some other shortfalls and how they vary latitudinally. Here we have phylogenetic information. So based on all those fully sampled phylogenies that we were using, we could track which species in those phylogenies were not represented by phylogenetic data. They were inputted. They were included in the phylogenies just at a random position at the genus level. So, and this happened for 1/3 of all tetrapods. And we can see that there is a huge latitudinal gradient here, showing that we are missing more phylogenetic information for species in the tropics, in global south mostly. And we took an opportunity of this database recently developed to explore what were the drivers of these Darwinian shortfalls, the lack of information on phylogenetic relationship across species. So we used several biological, geographical, and socioeconomic factors to understand the chances of a species having or not phylogenetic information available. And here we have what were the major drivers. I'm not going to talk about each one of those drivers. In the paper, we discuss this test separately across biogeographic realms, different spatial resolutions, but the major results, they started to point some similarities to us. So what we found, species that are rare, represented by a few specimens in scientific collections, species that are fossorial or arboreal, species that are small-sized with narrow ranges, they all have lower chances of having phylogenetic information available. In contrast, species that are large-sized, occurs in terrestrial or aquatic environments, have wide ranges, are commonly represented in scientific collections. Those species have way more chances of having phylogenetic information available on them. So this is pointing some similarities already across different shortfalls. And I'm going to talk about just one extra shortfall, and this is a shortfall overall. And what I would like to say by shortfall overall, it's actually the number of papers published per species. What makes a species highly researchable? So in this study, we quantified the number of publications available per species and try to identify what is determining this number. What are the drivers of that number? The map below, you can already see again a latitudinal pattern showing that species from temperate regions, they are better known than species from the tropical regions. And on average, we found 8.5 papers published per species. These numbers, they were all based in Scopus database. Tá? We found a median of 1 paper per species, so pretty drastic. 90% of the species have less than 10 papers published on them, and 40% have 0 papers, meaning that the only paper available for those species was the description of the species, right? And what were the drivers? Again, verticality. It's emerging here as an important factor, meaning that fossorial species and arboreal species have less papers published on them. Also, small-sized species have less paper published on them. In contrast, large-sized species, those occurring in terrestrial or aquatic microhabitats and threatened species by IUCN, they all have more publications available on them. So there are another drivers, including socioeconomic ones, that we explore in the manuscript. But I would like to highlight in this talk the similarities among some of these drivers. So we have talked about how to find new species, unknown species, using time-to-event models to identify species in the field or inside scientific collections. And we also have talked here about how can we quantify invisible fractions of the trait space and also understand common drivers of major knowledge shortfalls. It's important, I would like to highlight here, Johnny Getz. He's a former PhD student and he led 3 of these manuscripts. So thanks, Johnny, if you're watching. And also thanks to everyone else who collaborated with me in this research. And now we go to my final slide. So in the end, we have very different shortfalls. We're talking about shortfalls associated with phylogenetic information, natural history data, research effort, but we have similar drivers. And this is actually a good news, because if we have similar drivers for those shortfalls, it also means that we have a common solution. So it is important to better look at those species that are small sizes, those species that occurs in fossorial and arboreal environments, and also those species that are nocturnal. We have several commonalities here driving these knowledge shortfalls. And I guess here we have an opportunity to close those gaps more efficiently. So that's, I guess, everything that I would like to say. And once again, thanks everyone else who is watching, and I'm open to questions. Are you still here? Yeah, okay, I'm still online.
We are still here. Thank you so much, Mario. This was great. If you have questions, if anyone in the audience has questions, please put them in the Q&A box, and I am happy to read them out loud for Mario. So let's give folks a couple minutes to put their questions in there. I can kick us off with a question. I have many questions. This is wonderful. But okay, I'll start with a simple one. To what extent are those undescribed species that you talked about in the first part of your presentation? Are we talking here but are we talking here primarily about like taxonomic splits of species that we already have described? It's just that we've recognized through DNA sequencing or whatever it might be that they might be separate species. Or are we talking about like truly new species that no one has seen before? Do you have an idea to what extent, where is this balance?
We had some of numbers on that issue explored in the submaterial of that manuscript. I will not know that number right now, but I can anticipate that for amphibians and reptiles, these newly described species, they are above 90%. Mm-hmm. For mammals, it's nearly half or 60% of them. And for birds, it's less. It's like 10 to 80% of newly described birds are actually like a new population that was found. It's not a taxonomic split. But there are many splits going on for birds, a substantial amount of taxonomic splits going on for mammals, but almost no split is happening for reptiles and amphibians. Like when we look for the overall scenario, it's just a minor amount of those descriptions correspond to splits.
Yeah, I mean that makes sense. I mean that's what I would expect. That amphibians, reptiles are less known in general, therefore they wouldn't be, that truly would be new species. And so, we've talked a lot about tetrapods, and we have more data to make estimates such as you made for tetrapods. Do you have any idea how these numbers look for invertebrates, for example?
Oh, there is a plan to do this for invertebrates, but in order to do it, we need to actually degrade the taxonomic information. We will not be able to compute these models using species level data, probably genus level data, family level data sets. So we need to work at a higher taxonomic resolution to achieve those numbers for invertebrate groups, because at the species level, we have several morph species, so many undescribed specimens, and it's not possible to do it at species level. So we could, for example, degrade that information for tetrapods and compare how it goes. Okay, so we have to estimate based on a degraded data set, and we have an estimate based on a species level data set, and we can compare those and see if they match really well. Then we can estimate for invertebrates based on a degraded data set only. That's how I would do it.
And then make some sort of an inference how that would look maybe as well. Great. Okay, there's a couple of questions in the Q&A. The first one from Jean Pablo. How do you think these gaps in knowledge can affect our understanding of how biodiversity responds to environmental changes induced by human actions?
So, for example, linear shortfall, we are totally missing those species in any kind of conservation prioritization strategy. Like when we apply algorithms of systematic conservation planning or everything else, we are just quantifying the presence and absence of species out there. And this is done just for species that we already know. And for those species that we have information available on the occurrence geography. So when we do not include those unknown species, we might actually be saying that some region is unimportant because we don't know the species that are there yet, right? So it would be important to have more information available or be able to incorporate these unknown estimates as a weight, something like that, in a model of conservation prioritization. And also, there are some issues that have been pointed in the literature with how are we quantifying, for example, diversification rates? It's our metrics of diversification rates. They are based on the number of species known for a particular clade in a particular time interval. So when we are omitting those species, actually, it might be the case that we have way higher diversification rates in the tropic that we know nowadays.
Yeah.
We have a recent paper published in Systematic Biology, in which we do some exercises trying to understand how this would impact diversification rates. And there is also a huge latitudinal gap. There is a paper published in Trends in Ecology and Evolution called The Taxonomy Latitudinal Gradient, I guess. And they are discussing this idea that the taxonomic resolution of our information it follows a gradient where in temperate regions, the taxonomy of most organisms are well developed. They have gone through several different taxonomic revisions. You have well-established species over there. But as you move towards the tropics, like towards the equator, we have several species with higher uncertainty around them. We don't have like really exact numbers of number of known species for the tropics. So that can affect, for example, these estimates of diversification rates. And also other studies try to point how this affect the effect size of predictors. Like you might find the relationship to be linear, but it might also be exponential.
I think that's exactly what this question is asking about, because it seems that the question is asking about like responses to environmental changes. Like, do we think that species that have not yet been described for some reason might be responding differently to the same environmental drivers as a group than those that have been described? Like, do you think that if we start adding more species to our knowledge and start looking at how they, associate with different environmental factors, would we be finding differences than those that we already know?
If we have those threats spread in a heterogeneous way across space, probably we will have a different impact on different species, right? Because the unknown species, they are more concentrated in the tropics, near tropical broadleaf forests, right? And you can think about what are the threats impacting those species in those regions and how those threats are different from the threats that are impacting species elsewhere. Because we might be underestimating the impact of degrading tropical forests or harvesting tropical forests because we have not quantified the total biodiversity in those forests. So I would say that the impact is not important, just if all the threats, they were homogeneously spread across space. So that would not matter if they are affecting a described or undescribed species. But threats are not homogeneously spread across space. They are concentrated in specific regions. So I guess, especially in tropical forests, the threats that we are seeing in tropical forests, they are bigger than we are actually estimating.
Okay, thank you. Another question from Toby Pennington. Mario, great talk. I am a botanist. Any suggestion why the shelf life of plants in the study that you cited is the highest, higher than for animals?
I would say perhaps many of those, I'm not sure. I would say that plants might be collected by more often by non-taxonomists. That would be something leading to a higher shelf. That was the most important variable in our analysis. Like when a specimen was collected by a non-taxonomist, it had way higher chances of remaining hidden in a scientific collection. So I guess the collection of animals it's more atypical to be done by non-biologists, although it might happen, like farmers or environmental policy, they might collect some specimens and direct those specimens to a collection. But for plants, perhaps we have more people collecting plants and trying to store that information or donate a specimen, because we have, like, I don't know the name, it's exicata. When you put a plant in a collection, you can also make a art, a board for a wall. So they have different ways to, it's used as an art too. It's not just a specimen inside the collection. So perhaps we have more taxonomists working and collecting plants, more non-taxonomists collecting plants. I guess that would be my guess.
Mm-hmm. Okay, cool. A question from Elizabeth. Really enjoyed your talk. Given the challenges of gaps in knowledge at the species level, what is your opinion of species-based conservation approaches? Do you think there is a place for it, or should it be abandoned given how many species are undescribed or unknown?
No, I still think there is a place for species-level conservation approach. We need to, I guess there are other approaches to draw conservation prioritizations, like using phylogenetic diversity, phylogenetic richness, or functional richness. But we need to be able to sell our results to everyone else in the society and not just scientists. So I guess using species, it's an easy way to communicate with the decision makers, and it's still valid. I guess we need to work in multi-tax approach and try to include these estimates of knowledge shortfalls in our results. I guess that's what is important. We will not be able to close those shortfalls for everything. That's utopia, right? We will not be able to do that. But we can be able to incorporate those gaps in our analysis and understand what is the impact of those gaps in the results that we are expecting with conservation plans.
Okay. One other question from Sajat Nouri. Thanks a lot for your great talk, Mario. Do you have any idea if barcoding approach has been helpful for addressing these shortfalls and decrease the shelf life?
I have seen some research using barcoding to estimate like the diversity of specific regions, but I have not read that much on the topic. I guess to answer this question, I'm sorry.
It's fine.
But I saw some papers trying to use DNA barcoding to quantify the presence of species in some regions, and that's cool. I guess it's an important technique, but we need to have, because you need to compare like what you sample from the environment with a database in which you have the DNA information that matches with the one that you find in the environment. So if that database that we are using to compare our field samples, if that database is incomplete, the technique it's also been affected by a nodal shortfall.
Cool.
Right?
So just to kind of follow up from that, you've mentioned that, I mean, you've talked about the different drivers of potentially not being able to describe species, or poor estimates, species number estimates, and so on. Like we know more and more about what drives poor description, right? Rates of description. And you said, like, we need to look more for nocturnal species, and we need to look more for this and this, because we know that these are underrepresented in our databases. But how? How do you, like, what do you think should happen for that to actually happen, knowing that taxonomic experts, right, the taxonomists are a sort of a declining group, right? We have fewer and fewer taxonomists in museums now to be describing these species. So, like, how do you think we should proceed? Like, what should we do?
No, I guess looking for species that are, for example, nocturnal, this is like, we need to voice this message and hope that people planning to do field work, they also consider techniques that might involve sampling during nocturnal periods, or, for example, techniques that involve sampling fossorial species or sampling arboreal species. We have techniques that can be used to that purpose, like the place where we put in traps. You can put traps in trees. You can put like pitfalls on the ground to capture more of those specimens, and stop ignoring that thing. Because you can also do field work without using traps, without using pitfalls, without using arboreal traps, and you will get a result, probably underestimated result. But I guess my hope here is that people start to use more complete techniques to sample biodiversity. And I really hope that taxonomists working inside scientific collections, they could use the results of that second research to better explore scientific collections nowadays. We have probably thousands of undescribed species already collected inside scientific collections. They just need to be verified. So it's an opportunity. It's a huge opportunity for taxonomists with limited funding, for example, that cannot go outside and do field work in different countries. So they might look at these collections and rethink how they can process that material in order to describe these novel species.
Okay, cool. Well, if there are any last-minute questions, please put them in the Q&A.
Okay.
Can you hear me, Mario?
Yes.
Okay. I was worried
I
that
froze for a moment.
Okay, well, you seem fine now. But it seems like people have asked the questions they wanted to ask. I don't see any new popping up. So we can just wrap up a couple minutes earlier. It was an amazing talk, and actually gave me a lot to think about, because we can use this information that you talked about, right? Like, we can use these estimates of our knowledge gaps to correct our estimates of biodiversity, right? Just the way you did. So that's something that's food for thought. Like, how do we do that? How do we spread that message to scientists? Okay.
Yeah, we need to do a better job with journalists, with SciComm. I try to do a lot of that here in Brazil. That's why I hope that message will be spread beyond academia.
That's right. Yeah. And policymakers, right?
Yeah.
Excellent. Well, thank you very much. Actually, there's one more question. Since we have a couple more minutes, how about I read that? Fantastic presentation. Any comments or recommendations for working on marine fishes? Challenges from shallow to deep sea. That's a question from Adela Roa Baron.
So marine fishes. I'm not an expert on marine groups, so I guess if I wanted to recommend something, the first thing is, do we have like a global database of marine fishes? I guess FishBase it register marine fishes. So I guess the most problematic thing with marine fishes to run these models, in my view, it's actually to get information on socioeconomic drivers, because we have many of these databases reporting human population density, GDP, whatever, all this information, they have been available at the country level, at the city level, or for terrestrial environments. We don't have that information available for marine environments, but if you are working with coastal species, perhaps you can expand that information, derive it from distance from the coast, and get the closest territory to get an information of socioeconomic. But I'm still unsure how it will be representative for the coastal fishes, but that's on our way out. And I guess it's a bottleneck to process the data for marine fishes. It's to get information on socioeconomic drivers that are really important. You cannot let them out of the model, but other stuff, it's really general across taxa. You can have information on body size, range, heredity, number of taxonomists working with the group. Like if a species is nocturnal, diurnal, perhaps, I don't know if that's possible to get for fishes, but I guess biological and taxonomically related metrics are easily derivable for any taxonomic group, but socioeconomic metrics will be most often connected to spatial information. And if the species is occurring in high sea, probably we will not have a way to incorporate socioeconomic drivers in your analysis. I guess I would think a little more about that.
Excellent. Well, thank you very much, Dr. Moura, for joining us for July Funk lecture. It was a great presentation. I'm sure everyone has enjoyed it tremendously. I know I have. And thank you to all of you for joining us again for our seminar series. Please keep coming. We wouldn't be able to share this wonderful research with everyone if it wasn't for our audience. So please consider coming to future meetings as well. And just as a reminder, we will be meeting next in September. We'll be skipping August. And in September, we'll have Brian McGill speaking to us about some of his research, which he's done plenty of. And I'm sure it will be a wonderful talk as well. So thank you again, Mario. And till next time.
Bye, everyone. Thanks
Bye,
for
everyone.
watching.